Good afternoon. Today I'll be talking about my breeding program at Louisiana State University. It's a rice breeding program, and in the last couple years we've started to move towards integrating molecular markers as a key component of these breeding activities, and we've worked very closely with LGC setting up our lab, and a lot of challenges and hurdles we've come to during this process. We've worked very closely with LGC, been a very good partner, and I appreciate the opportunity to come here and speak to you today. Okay. So I'm going to talk a little bit, just a brief introduction to rice and some of the objectives of the breeding program, kind of the structure of our breeding program, and how that impacts how we can use markers, and also where markers can have a beneficial contribution. We talked about some of the logistical challenges and considerations when thinking about utilizing technologies and markers in an applied breeding program, and then give some examples of how we're actively using markers in our program today. Okay, so first, rice is a relatively small crop in the United States. It's grown in 5 southern states. We're here in Louisiana and in the Sacramento Valley in California. Unique thing about rice, I guess, compared to other big crops, corn and beans, is quality is a very, very important component. Also in the United States, export market is very important for rice. So over 50% of the grain is exported, and the quality parameters are very important thing to consider. It's just acreage in Louisiana last year, and this is just to show all the lines here that are circled. These are varieties that are developed in our program. So the work we're doing is very focused on the farmer, focused on productivity in the field. So not just one trait, it's the whole package of traits. There's about 25% of the acres are hybrids from Rice Tech, but about 75% of the acreage in our state are varieties developed within our program. So that's something to keep really focused on, is that end goal of developing a product that's going to work out in the field. We work at a research station about an hour and a half from our main campus, and we do very applied research and extension activities. All of our funding, I shouldn't say all of our funding, but a lot of our funding comes from the Louisiana Rice Research Board, which is a checkoff fund-based organization. So this is money coming directly out of their pockets. So we want to spend that money wisely and in a way that's going to bring short to medium term returns for their profitability. So I'll talk a lot through this I talk today about some of the logistics and costs and things like that, because at the end of the day, we really have to spend these farmers and growers money wisely and make sure that what we're doing is actually going to have a return on their bottom line. So the station has been there for over 100 years, and the station started around this time, and in that time, rice yields in Louisiana have kind of just continuously gone up. We're around 7,000 pounds per acre right now. It's a tough place for rice to grow. It's very hot, a lot of disease, very humid. Our varieties do much better if you get a little further north into Arkansas, but the program has been successful, and we've driven gains continuously, and we're hopeful that the next step, or to help continue these gains, that the markers and new technologies will help drive that. The breeding program really has two primary objectives, or two primary targets. The first is yield, and the second is quality. Quality in rice is a complicated trait because there are many different aspects of quality, from cooking, appearance, texture, chalk, milling. So there are many traits under there, but these are the two things that at the end of the day, the industry and the growers care about. Besides those traits, though, we have many other traits that we have to focus on that have impacts on quality and yield. We have 3 different segments for herbicides: conventional, non-herbicide resistant. We have clearfield rice, and we have a new different mode of action, Provisia rice. And then we have some major diseases we focus on, insects, agronomic traits. And I just raised this point because there are many traits that we have to focus on. It's not just one or two traits, and we have to keep all these things balanced. And the markers are giving us an opportunity to push some traits and maintain other traits. So this is a figure of some of the key traits that we're focused on, and basically it's just a relative thing, traits one another. But on the y-axis is our capacity to phenotype, which to me is how cheaply can we do it, how fast, how accurate, kind of composite of those things. And then on the x-axis is the genetics of the trait. So traits that are down here, our phenotyping capacity is relatively low, and the genetics are complex. And traits that are up here, we have a high phenotyping capacity, and the genetics are simple. So as you can imagine, the traits up here are a lot easier to breed, a lot shorter time frame, and a lot higher probability of success. Whereas you get down here, these traits are a bit more complex, much more investment required to maintain those gains. So this similar slide as James had in his previous talk, but this is a general kind of our breeding funnel. And we start out with single plant selections. This doesn't matter if you're doing markers or just phenotypic field-based selection. We'll do single plant selection, and there's hundreds of thousands of plants, and then we'll go to head rows or progeny rows, and then yield testing, multiple locations. So the numbers of entries as we move through here are going down pretty much by an order of magnitude. And then simultaneously, the investment we're putting into evaluating each one of these experimental entries is going up significantly. So at this early stage, it might be as simple as walking by and is it not too tall, or is the maturity correct? Whereas these later stages, we're testing across 5 states, 15 locations, replicated trials, chemical traits, disease nurseries, all sorts of things. So the investment on an individual line is significantly more as we move through this funnel. So this should look familiar as well from James's talk. What are some of the key factors for progress? And again, this is just a general breeder's equation. It doesn't matter what approaches you're taking, but at the end of the day, the breeder will typically think back to this. We're focused on our end product, the variety, and how it's going to perform in the field, not so much the technology or the approaches we use. So a few things, the number of experimental lines evaluated. The more we look at, the more gain we're going to have. But we can't sacrifice the accuracy of the data, so that's going to be a function of your heritability, how accurately you can phenotype, the amount of genetic variation. I put useful genetic variation. So the more variation we have, the more gains we're going to obtain. And then the speed of the breeding cycle on the bottom of this equation. The faster we can recycle elite lines as parents back around, the more progress we're going to have. So that's a general equation I think most breeders kind of think back to when they're structuring their programs. So go back to this little figure with the traits that we're interested in. And so what are some of the breeding constraints? So as I mentioned, in the early stages, we're looking at hundreds or tens of thousands of experimental entries, but our investment is relatively small into each one of those entries. So we're pretty much limited to these traits that we have a very high phenotyping capacity for. Herbicide is very simple. You spray it. If it lives, it's good. Grain shape, we can kind of walk by and observe, does the grain look to be correct before we select. Height, very simple to look at. Maturity. So these are kind of observational traits. And we're constrained because at the early stages of the breeding program, these are the only traits that we can effectively select on with the numbers of material we're screening. I mentioned before the quality traits. These are the 2 major quality traits, chalk and milling, and yield. These are the traits at the end of the day that we really care about. And you see these are kind of in the more difficult space of our breeding. And so we don't really begin evaluating these in any meaningful way until we get into this stage of the breeding program. So the number of entries that we're looking at, it's not really hundreds of thousands of lines that we're evaluating for yield and quality. It's more in the range of a couple thousand. So we're limited on the number of lines we can evaluate for these complex traits and the traits that we're ultimately interested in. So when we think about molecular markers in the breeding program, how can they help us? And you think back to that breeder's equation and the things we're trying to do. Well, one, the markers allow us to initiate selections earlier. So if we have markers, I won't talk about this, but for types of predictions and things like that, instead of waiting for yield and quality till we're at this stage, maybe we can start it back here, and the number of lines we're effectively evaluating for those traits goes up, and that has a very beneficial impact on our gain. The other is increased accuracy of the traits. So some traits, actually the marker is more accurate than the actual phenotype. I'll give an example later of a disease trait, but that's an easy one. But the markers often will take into consideration multiple years of data. So even if we had excellent yield data from 1 year, or from the field for a given trait, it's only from 1 year, where the markers take into consideration multiple years. And the last, you can reduce cycle time slightly, I guess, but a lot of this time in the breeding process is really dedicated to testing, which is something we're always going to have to do, is multiple years of testing before variety release. But you can maybe trim a little bit of time off the cycle where you can recycle something back to be a new parent. So, like I said, our program, we're just now beginning to implement markers as a kind of a core piece of the program. Rice has been a model species. There's been a lot of knowledge for decades in rice, but there's been relatively little utilization of markers in applied breeding programs. There's been a lot of activities such as backcrossing and things like that on a trait-by-trait basis, but widespread implementation has been limited, and I think there's two main barriers to that. One is cost. The cost per data point was prohibitive in that even if you had the money, you still would have to take a step back and say, could I spend my money better by putting more locations or things like that? So cost was a limitation. And also DNA extraction has been a limitation, and both in cost and just in time. And that's the other thing related to throughput. The number of samples that could be processed through a lab with say, gels or SSR type of technology was limited. That yes, you could do it on a handful of populations, but in the scale and the size of what we're doing, a few populations is just a drop in the bucket of what we're trying to do. The other is the turnaround time was relatively, it's too slow to make breeding decisions and move material. So those would be, I think, the two major barriers have been. And when we started looking at opportunities, we shopped different options and determined that the LGC SNPline system was a good fit for what we're trying to do. You see, this is our lab. It's literally, this is all the equipment. It's not very fancy. It's not very big, but for our purposes and the objectives and the types of activities we're trying to conduct, it works very well. So this system is very useful for us. It's appropriate for breeding applications where we have many samples and relatively few markers. So most of our activities don't require whole genome sequences. Most breeding activities don't require that level of density. So we are doing about 60,000 samples a year. And I just estimated this, but probably over 95% of the data points that we generate are from less than 30 different SNP assays. So this system is very good for that. System also works, we do a very quick and dirty kind of a hotshot type of DNA extraction. This is important because if we're doing $1 extraction or something like that, it's $60,000 a year in DNA extractions, and that's a good chunk of what we have allocated for this marker lab. So we're doing a very cheap and dirty DNA extraction, and the system works pretty well with that. The throughput is sufficient for what our needs and objectives are. One person can run 36,000 data points in a day. So the throughput in the lab is never our bottleneck. Our biggest bottleneck always is it's not cost, because the cost per data point, we have it down below 4 cents. The biggest bottleneck and what we always plan around is collecting the tissue. So if anyone has a good idea or a good system to how to collect just the leaf tissue in a high throughput way that's also relatively cheap, that would solve a lot of our challenges. So what are some of the applied breeding applications we're doing? Now this is relatively basic stuff. It's nothing that would impress anyone as far as the technology, but I think a lot of this just goes back to logistics and some of the practical aspects. So one is a big activity is marker-assisted selection, forward breeding for traits of large effect, back-crossing new traits in. This is useful in that, like I said, rice quality is a very important parameter. So introducing novel, say, resistance is very difficult, because when you bring in that novel resistance, you also bring in a lot of undesirable quality and traits like that. So the system is useful for that. Some more kind of simple things, F1 cross characterization. All of our F1s, we genotype with a small set of panel SNPs that we've developed that allow us to first ensure that the cross was successful, wasn't a self, and also that the right cross was made, or something didn't get mixed up. The other big area that we do a lot of data points in is foundation seed purity. We produce and have a foundation seed program on our station. So all of the head rows that go out in that foundation seed, will do usually about 10,000 samples a year, just genotyping those to make sure that something didn't accidentally outcross or get mixed up, or a weedy rice get in there. So these are things that have really transformed what we can do. These are a picture of people going and pulling off types from a foundation field. The amount of that we observe now just from genotyping these foundation seed lines, it's drastically gone down just in 1 year. You see the purity and the heterogeneity within our lines has gone down. So again, not the most scientifically exciting thing, but a lot of practical applications, and it really has a lot of impact on what we're trying to do. Okay, so I'm going to shift and talk about kind of the integration of the marker selection into the breeding pipeline. So this is a general breeding structure. It doesn't matter if you're doing phenotypic selection or incorporating markers. So in its early stages, where we're looking at single plants, this is where we focus our marker-assisted selection on. We typically do about 2 rounds of this a year, somewhere between 20 and 30,000 samples each round. And here we're focused on single genes or loci of very large effect, qualitative types of traits. So this is a map of, these are all the different KASP assays that we've designed and validated in our germplasm, and don't worry about going to read those, but we probably have about 20 different loci that we have markers for. Some of these loci I'd say are ideal for marker-assisted selection. It's kind of a 3-to-1 Mendelian type of trait. Some of them, my opinion is they're probably not the best targets for marker-assisted selection, because they might only explain a portion of the overall variation. But I put this up here to say, okay, there's many traits, there's many others. There's probably more than this discussed at this conference. But there's some practical considerations when we think about doing marker-assisted selection. One is just the number of loci. You could have 20 different genes, but it's not very practical. It's not very practical to select on that, because if you have one gene and you're trying to fix it, 25% of the plants. Okay, so that's pretty good. We typically strive to come out with 100 selected lines to go to the field with after marker-assisted selection. So if I'm just fixing one gene, okay, I have to genotype 400. That's pretty easy. But if I say I want to fix 8 genes, I'm going to need to genotype 6.5 million plants, which is not very practical. So another strategy we take sometimes, if there are multiple genes, is just to enrich them. But basically, I just highlight that there are many, many genes out there, but it's not as simple as someone saying, oh, I discovered this gene, here's a marker, you should use it, because there are other traits you still have to select on and prioritize. The other is a logistics around just the configurations and cost. So we found we use a 1536 format for all of our PCR, and basically found that it's certainly the cheapest, and if we plan ahead and plan out our molecular breeding activities, we can get everything to fit into that format. And you see we have a data point cost of 3.7 cents if you're doing a 1536, but if you do it on a 384 basis of 4 384s, it's almost triple the cost. So a lot of planning. Again, it's not rocket science, but it's just a lot of logistics and planning of how to fit all these things together in the most cost-effective and time-effective manner. So the example I'll walk through is blast is a disease. It's a fungal disease. You can get on the leaf, but also on the neck, and this is where it's a bigger issue. You can get sterility of the panicle or the head. So if you look at where blast falls on our little diagram of our traits, it's a great trait for marker-assisted selection because it's relatively simple genetics, single genes, and it's a very important trait as far as for the market. And this is just a picture. These are susceptible lines in our blast nursery and resistant lines. So it can literally kill the plant if it's bad enough. But I show this picture as well because we can phenotype, but we are limited. You have to grow it out in a special nursery. So we are limited to a few thousand lines of what we can actually put to phenotype. So the markers allow us to certainly increase the number. The other thing the markers are allowing us to do, we have different blast genes. Each one of these is a different gene. And these are the different races of blast in the southern U.S. So we know that, okay, this gene here gives resistance to all these races, whereas this gene only gives resistance to these. These are things we wouldn't be able to detect very easily in the field. So the gene I'm going to give an example of is this gene called Pita. It's this very common gene people working in rice would be familiar with. It gives very broad spectrum resistance to blast, and it's been deployed globally. So what we typically do to develop and validate our trait markers, we'll characterize all the haplotypic diversity in our germplasm. In our germplasm at this gene, I think there's about 5 haplotypes, but 3 of them are at extremely low frequency. So here I just show the example. If you have this haplotype, 57 of our lines in this collection you have an average score of 1, which is quite resistant. If you have the other haplotype, you have an average score of 5.2, which is quite susceptible, and actually a lot of these lines down here just have another gene, and that's why they're here. So this is a great target for marker-assisted selection because it's a very large effect gene, very well characterized. When you look at this gene just in a biparental, this is a set of rills. It's almost perfectly correlated, and sure, some of these that are off are probably just a phenotyping or a seed issue. So it's a pretty much perfect trait as far as explaining all the variation. So it's an ideal target for marker-assisted selection. So when we go back to thinking about where the markers fit in this scenario, we can initiate selections earlier. So we can start doing very effective blast selections at this first stage in our breeding program, whereas we don't start disease nursery screenings till here. So that's an advantage to take the marker approach for this trait. We have an increased accuracy of selection with the markers compared to the field because we have different degrees of pressure of blast year to year. It's also someone, a pathologist, we have an excellent pathologist, Don Groth, but he's just walking through and rating them. So the markers, I have a lot more confidence in the markers on a large scale than a single year's data in the disease nursery. Increased number of lines screened. Our blast nursery, we can only handle a couple thousand lines in there, whereas we can handle basically tens of thousands through the markers. And the other thing is we can select on which blast genes are present, not just the phenotype we see in the field. So it can maybe help combine these genes in a favorable way to increase the durability or the spectrum of which they confer resistance. So our workflow is relatively simple, but again, it's just the logistics of this are our biggest challenge. So the way we do this, we'll typically do what we call a batch. So we'll do a batch of marker-assisted selection a week, and we'll typically do 32 plates, so about 3,200 plants. And we just plant them in these little flats, and this flat has the same layout as a 96-well plate. So we don't need to worry about tagging plants or labeling plants. It's the plant's ID it happens to be in whatever tray 30A6, and that would correspond to the DNA plate. We'll take pieces of leaf from here a week after planting. We have this little punch, and this is a kit that LGC provides, and it is very useful to get uniform sizes of tissue. And like I said, this is the biggest limitation of what we do. When we plan everything, we say, how much time do we have? How many people do we have? How much can we sample? And everything else is pretty straightforward. So we'll sample into these plates, we'll extract in these plates, and we'll do PCR basically one step out of these plates. We'll go through our lab, which we have on station, and about 2 weeks after we plant these seeds and a week after we sample them, we hope to have the data back. Most of our trait markers, we do a good bit of work to try to get them to look. They don't always look this good, but something like this where you get very nice clear clusters and high data return. And then for our selection, I mentioned one trait. Let's say we wanted to fix it. You need 400 plants. Well, we're not going to do 400 plants because that doesn't really fit well in the plate. So we'll do 384. So my goal is always come out with 100 plants, and that's just the way our field's set up for head rows. We do 100 rows for a family. So if I have 384, you get missing data, I'm not going to end up with 100 plants that are fixed. So through a few rounds of doing this, what we've decided and we've come up with is we say we'll put things into groups. So group 1 might be the most desirable group. And now we don't keep track of lines anymore. We say, okay, this has the right quality of these 2 traits, and it's homozygous resistant. So those lines might be group 1. And one population might have 20 lines in group 1, another one might have 80 lines in group 1. And then we'll go through with a group of people and we'll say, okay, flat 22, and they'll just read A1, A6, B12, and we'll just pluck those plants out. And at that point, we don't worry about keeping those plants separate. They'll just go in a bundle and they'll get planted into either pots if it's not a good time to go outside, or it'll get transplanted into the field, and then they'll just be called group 1. Group 2 is this, in this case, it's het at this gene. It's the second most desirable, and group 3 is the least desirable. And then once we grow these plants, we'll grow them out, we'll get panicles, harvest the seed off those panicles and go to panicle rows. And the way we plant those in the field, as when you're in the field making breeding selections, you don't even need to know or care about what markers were selected. All you need to know is group 1 is most desirable, group 2 is second most, and group 3 is least. And we'll plant these out, group 1 first, and then we'll put a purple marker, and then group 2. So actually when we're doing our phenotypic selection in the field, most of the time I don't even look to see what this breakdown was of what defines a group. We just know I want to take as much from here as I can, and then here, and then if I have to from here. Okay. So with that, I'll just summarize here. A lot of what we do in an applied program where we're really not so interested in the technology we're using, it's more about the end product, is resource allocation. And I think now with the markers, that is starting to shift where it actually does make economic sense or resource allocation sense to shift some things towards markers away from the field. Trait targets really need to be focused, and the research investments must be justifiable. Like I said, we get all of our funding from the rice growers in the state of Louisiana. It's their personal money, so we really try to stay focused on delivering what's going to be best for them. If I know here, everything is a trade-off. So if we work on an additional trait, that's that much less attention we're putting on yield and quality. So as people come and say, hey, you should work on this trait or think about that trait, we have to say, okay, well, should we take focus away from yield or take focus away from quality to work on that trait? Technology offers many opportunities to increase our productivity, but we have to be very careful that it doesn't become a distraction to what we're ultimately trying to do. I should also say, the program that I work in now, and I've just been there for just over 2 years, but inherited a very sound program in terms of foundation and just basic breeding infrastructure and field techniques. So it's a good program to build this on top of, but I'd say if a program doesn't have the basic infrastructure in place of just the fundamentals of breeding, maybe these technologies might be putting the cart in front of the horse. Marker technologies are becoming cost-effective and practical for significant integration into public programs. The big companies have been obviously doing a lot of this large-scale marker molecular breeding for a number of years, but they obviously have some more resources to do that. But with companies that you can work with, like LGC and technologies advancing, we feel as if this is going to become more and more of a reality in public programs. And finally, it's critical to work out the logistical aspects to ensure that the technologies do not hinder the program. So I showed that slide earlier of the progress in yield in Louisiana. It's being relatively new there. If I don't do anything different, I assume that trajectory will continue. So you don't want to change too much too soon. So working out a lot of the logistical aspects, and it's a lot more of an issue or a lot more challenging than I would have ever imagined. And any one thing it's not It's not rocket science, but it's just a lot of things on top of each other while still keeping the main program going. So I think the biggest hurdle for public programs or relatively small programs going forward is working out a lot of these logistical things while still keeping the program moving forward. So with that, some acknowledgments. Just want to thank all the project leads at the Rice station I work at. All the different programs work very closely and contribute to the breeding program. I have excellent staff. We have a few people who oversee a lot of the marker work and lab work, and then field staff and some graduate students. And then these are a few people I work with on a very regular basis at LGC. And like I said, LGC has been an excellent partner for me as far as I go with them with problems and say, hey, you guys really don't offer this. Is there a way we can work something out? And we've always been able to come to some solution. So having a good partner like LGC has been very critical for us to kind of get where we want to be with this. And last, I just want to thank our Rice Board again for the funding. They support us very well, and we appreciate their support and try to do our best to use their money wisely. So with that, I'll wrap up. If there's any questions, I can maybe take a quick question or pass it on. All right, thank you.